So here we find ourselves in Chapter 17. Chapter 17 is all about adaptive immunity, the immunity that we develop as we encounter antigens in the body and then mount an immune response to it. Susceptibility, and we'll start just by defining these terms. Susceptibility is the vulnerability of a host to being harmed by an infectious agent. If you are susceptible to the disease, that means that you could get the disease. Immunity, on the other hand, is the idea that we can recognize when an infectious agent comes around and mount a defense against it of some sort. Adaptive immunity is this ability to recognize a particular infectious agent and then be able to respond to that infectious agent and develop the ability to fight it off again much more rapidly the next time we run into that infectious agent. If you study adaptive immunity, then you're involved in the field of immunology, because that looks at how the immune system responds to specific infectious agents and to the toxins that they produce or that can be introduced into the body. Last of all, the immune system is the cells, and especially the lymphocytes, and the organs. The thymus would be an important one, the spleen is another important organ, that all work together to help to provide the host with specific immunity. So as we've looked already, there is the idea of innate immunity, or genetic immunity. That's the immunity that individuals are born with. And it's a genetically determined characteristic. And it includes species immunity, the fact that there are some infectious agents that can cause disease in plants or in other animals, but they don't cause disease in humans. So Bacillus anthracis, that's the disease anthrax, or that's the causative agent for anthrax. It causes disease in mammals and birds, but not in other animals. So reptiles, or other reptiles besides the birds, so things like lizards or crocodiles or salamanders or frogs, they don't get anthrax. Canine distemper causes disease in a bunch of different mammals, but it can't cause disease in humans because it doesn't have an important receptor that allows the virus to adhere to any of our cells. And Mycobacterium avium, it only causes diseases in humans if they have a severely depressed immune system. So people who have HIV, who are HIV positive and have developed full-blown AIDS, then their immune systems are depressed, and so Mycobacterium avium can cause a disease in them, but not to those of us who have relatively little health concern. So species immunity is one important piece of innate immunity. The other important piece is that with innate immunity is that it comes with the ability to recognize a non-self threat, so a non-self cell, from the self cells. And we've talked a lot about that in Chapter 16, so I'm not feeling like I need to go much further about that. On the other hand, adaptive immunity is the immunity that we acquire when we inherit, in ways other than our genetic inheritance. So it's when we acquire diseases because we encounter them in the environment, or they're spread to us, or we get a toxin introduced to us, and we mount an immune response to that. And there are two ways to get adaptive immunity naturally. So if we follow our flowchart here, this is the large topic of immunity. There's the innate immunity, what you're born with, and then there's acquired immunity or adaptive immunity. And we can get that adaptive immunity either naturally, or we can get it because it's been induced artificially. And so there are two types of naturally acquired immunity. There is naturally acquired active immunity, and there's naturally acquired passive immunity. With naturally acquired active immunity, antigens, these are non-self molecules, so molecules that don't have the markers that indicate they belong in the body. Antigens from the environment enter the body and trigger an antibody's production and other kinds of specific defenses. So exposure to an infectious agent is naturally acquired, didn't mean for that to happen, is naturally acquired active immunity. But we can also have naturally acquired passive immunity, and this happens as antibodies are transferred from a mother to a fetus. That's really about the only way you can naturally acquire passive immunity. So passive immunity is just the idea that you are delivered already made antibodies, and the only way that you can get them naturally is from the mother. Okay, so acquired immunity, naturally acquired, and active because your body mounted an immune response and made its own antibodies, or naturally acquired passive immunity because your body was delivered these antibodies. There's also artificially acquired immunity. This is the kind of immunity that we get when we are delivered either a vaccine, or if it's artificially acquired but active immunity, then once again we make our own antibodies. So here if we follow it, immunity, acquired immunity, actively acquired immunity, and if it's artificial, it's because we had some antigens delivered to us either as live antigens, and if they are live, they would be, the term that we use is attenuated. Attenuated means that they've been weakened so they can't actually cause disease. Or we can have antigens delivered to us in the form of a dead organism with all the antigens on it. Or they can break it down into the individual bits that are just the antigens. Or you might get a toxin delivered, or a portion of a toxin delivered. And in response to that, the body triggers antibody production and antibodies against that antigen. So we make our own antibodies, but we make them against a vaccine delivered. That's artificially acquired active immunity. If, on the other hand, we're just delivered a whole bunch of antibodies, here, take the syringe, poke it into your skin, and press the plunger, and now you've got gamma globulin shots, for instance, like ROGAM, or snake antivenom is another possible antibody vehicle. Those would, so that would deliver antibodies, they would do their antibody job, but over time, because we didn't mount an immune response to them, over time those antibodies are going to be destroyed as they age out. So immunity, when it's when it's passive immunity, it only lasts for weeks to months. So passive immunity is short-term. Active immunity is what gives us long-term disease resistance. So what is an antigen? Well, I've been using that term a lot. An antigen, sometimes called an immunogen, God, I can't say that. Let's try that one last time. Immunogen. Antigens are substances that the body and the immune systems of the body identifies as being foreign and causes an immune response. And usually these are large proteins, but they can be polysaccharides, or they could be a sugar and a protein together, a glycoprotein, or nucleoproteins, so like a protein plus a nucleic acid. Antigens are determined by DNA. Because they are proteins, they're determined by DNA, and they're found on capsules, they're found in the cell walls of bacteria, in membranes of bacteria, they're found on the envelope or in the capsid of virus particles, or if it's a protein venom, say that a spider or a snake has delivered to the body, then it's those proteins that make up the venom. And bacteria especially, but bacteria and viruses both, will have multiple antigens on them. Or the antigen may have multiple antigenic determinants, sometimes called epitopes, the region of the protein that determines or that triggers immunity. So here we're seeing that we've got the green, each of these that has only one, that has only one. Those guys are the same antigen. Here's another one. Let's see if there's another one like that one. So you can look around. There's a bunch of different antigens that are found extending off of this bacterial cell. And so lots of antigens, and different spots on them may act as antigenic determinants, may trigger immunity. So small molecules, like they're called haptens, frequently they're unable to trigger allergic responses on their own. But if it can bind to a larger protein in the body, then that complex, the hapten plus the larger protein, that can form a complex that is the antigen. Oh, here we are. See it looking at, okay, so here's a protein, and here we can see all the different epitopes, the antigenic determinants that are associated with that particular protein. Antibodies, these are proteins that are made in response to an antigen. And it's this odd little Y-shaped kind of thing. I should probably draw it better. Let me make this. So it's got a heavy chains, two of them, and then the light chains, two of them as well. And so these are proteins. So each of them is a string of amino acids. And they have the capacity here at this end and at this edge, they have the capacity to bind to the antibody, or to the antigen, I'm sorry. The antibody binds the antigen, and then that likely will trigger cell processes that result eventually in inactivation of the antigen and its removal from the body. And when we talk about how much antibody there is in someone's blood or body fluids, that's described as the titer. That's the concentration of an antibody that is required in order to bind to and inactivate a specific amount of antigen. So this is a measure of how much antibody there is in the blood, is the titer. And it's whatever, the amount of antibody that it takes to bind up and inactivate some known amount of an antigen. Okay, so what are the cells that are associated then with the immune system? Well, there's two primary types, and both of them are lymphocytes. So lymphocytes are the cells that are involved in providing specific immune response. And they, like all the other white blood cells, develop from hematopoietic stem cells that are found in red bone marrow. And there are two types of these lymphocytes. They're the B lymphocytes, sometimes referred to just as B cells. And they are the ones that differentiate and mature in bone marrow. And bone marrow is what is considered a bursal equivalent tissue. So if you saw that picture of the chicken in the book, this is the reason why the chicken is in there. Because when B cells were first found, they were found in birds. Birds have this little sac right near their cloaca. That's where the digestive system and the reproductive system and the urinary system all come to a common exit. And so right near that cloaca, there's this bursa of Fabricius, and that's where B cells differentiate in birds. We don't have a bursa of Fabricius, but we do have bone marrow that acts in the same way as the bursa does. So that's where B cells come from. For me, I like to remember B as bone-derived, meaning that they come out of, they do all of their development in red bone marrow. B lymphocytes make up about 10% of all the lymphocytes that are in circulation in the body. And we find them in lymphatic tissue throughout the body, including the lymph nodes, the spleen, tonsils, adenoids, the galt, which is the gut-associated lymphoid tissues, including the appendix and the Peyer's patches that are found all around the intestines. So those are the B cells. T cells, they are, or T lymphocytes, these guys differentiate in the bone marrow. So they grow from that hematopoietic stem cell in the bone marrow, but before they are fully mature, they leave the bone marrow and they migrate to the thymus gland. And once they're in the thymus, they then finish their maturation process. And they make up about 75% of the circulating lymphocytes. And they're seen in all the same places that we see B cells. So they head to the thymus. That's the place where they mature. There are a whole bunch of specific hormones called thymosins that help direct their maturation. So B cells and T cells, both very important to specific immune response. And here we see a little bit about all of this development of the cells. So here we have these stem cells. Some of them go to the thymus to develop into T cells and then find their way to a lymph node. Others are just remaining in the bone. They develop into B cells and then find their way to the lymph node. So when activated, the T cells do some very interesting stuff. First, when they get activated, and by activation I mean when they learn to recognize a specific antigen and become tuned into that antigen only, they will divide and divide and divide, and their offspring cells will differentiate into a number of different cells. They differentiate into cytotoxic T cells, to delayed hypersensitivity T cells, helper T cells, and regulatory T cells. And we're going to look at these eventually. So following their production, then these cells migrate among the lymphatic tissues, and they use the blood, and they will maybe even go into other tissues as well. And each one of them has a different important role. There are some other lymphocytes. So if you did your math back there and saw that B cells were 75% and B cells are 10% and T cells are 75%, that only is 85%. So there are some other lymphocytes out there. One of them is the natural killer cell. If I could be any cell in the body, this is the cell I would be, because first they have the coolest name ever. And I don't mean NK cells, that's dorky. But being a natural killer cell, I like that. It sounds like I'm a spy. So, and then they also have the capacity to administer what, in one of the books when I first started teaching anatomy and physiology, we used a book that had a teacher's guide with it, and so I was trying to learn how to teach all this, and so I was looking at the teacher's guide, and they talked about natural killer cells as being able to administer the kiss of death, which I thought was pretty sexy sounding too. Hey, I wouldn't mind having a kiss of death. But when they run across a cell that has been infected with a virus, and cells that are virus infected frequently put up specific markers on their membrane, or if they find cells that have become cancerous, then they approach that cell and they release cytotoxic molecules called perforins, and these perforins will poke holes in the cell's membrane. And so there's the kiss of death. They come up close and then they squirt them all over with perforins. And once the perforins poke holes in the cell, then the cell contents leak out and it dies. They also send other molecules into the cell that will fragment, break up the DNA, and they cause programmed cell death, also called apoptosis. It looks like apoptosis, but it's not. You say apoptosis, so the second P is silent. And that is just a relatively organized kind of suicide of the cell that involves the lysosomes all breaking open and digesting itself. Okay, so there are two kinds of immune responses that T cells and B cells can trigger. There's humoral immunity, And that word humoral, that doesn't mean ha ha ha, that's so funny. Instead, it's an old school term for body fluids. So in Shakespearean times, they talked about, choler and phlegmatic and sanguine, and these were all the different body fluids. Choler was like a liver secretion that came out with fecal material, and phlegm, of course, that's phlegmatic. So those are the secretions coming out of the nose and the throat. And sanguine, that means blood. And so all of these humors had to be in balance in order to be healthy. So we wind up using this term humor in present day to mean body fluids. And so humoral immunity is all the processes that lead to the development of antibodies that are circulating in blood and in tissue fluids. So antibodies, they're found just running around our bodies, in our blood, in our tissue fluids. And so they're in the humors, and therefore they are humoral immunity. And primarily, humoral immunity involves the activity of B cells. And they defend against the body against foreign materials that are outside of the cell, because that's where the antibodies are found. And this includes toxins and bacteria and viruses before they get into the cell. All of that is going to be taken down by humoral immunity. On the other hand, there's cell-mediated or cellular immunity. This is the work that T cells do. And these act against antigens that are found embedded in cell membranes or that are found inside the cell. And so they defend against virus-infected and cancerous cells. They defend, the T cells do, against eukaryotic parasites, and they will also attack transplanted tissues. It should say tissues here. So anything that is cellular, it could likely be taken out by T cells. Anything that involves stuff that's just found out in body fluids, it's going to be taken down by antibodies made by the B cells. Okay. All right, so there you have it.